This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on April 9th, 2020. I'm Vincent Dracagnello, and you're listening to the podcast all about viruses. My guest today is the Director of the Institute of Virology at Charité Hospital Berlin, Christian Drosten. Welcome to TWiV.
Thank you. Hello.
Good to have you. I really appreciate it. I know you're a busy guy. As a coronavirologist, you're very much in demand these days.
Yeah.
And but we have known each other for a while. In fact, I had you talk at an ASV a number of years ago, right?
Exactly. And that was about MERS.
Yeah. And now we are back with another coronavirus. So I thought we could do a deep dive into this today. Mm-hmm. And I want to just mention at the outset, you have started a daily podcast, which is Das Coronavirus, right?
Yeah, exactly.
What a great name.
Update.
So you do it daily, right?
Well, it has been daily up to now, but I will decrease the frequency because it's just not doable anymore. To read along and go through all the preprints and so on. What I try to do is discuss preprints, but it takes more and more time to filter through them.
Yeah, we do the same thing on TWiV, of course, and we have increased to 2 or 3 times a week. But we do 2 hours per episode, so
an hour, yeah.
It's a lot. So, do you think you will continue that beyond the end of the pandemic?
No, I don't expect that. But I think it's a useful thing currently, probably one of the more useful things that you can do as a virologist working on coronaviruses in the present situation, because it's clinicians and, I don't know, many other disciplines actually doing this acute work that's necessary now. Yeah.
Of course, on TWiV, we always have said that scientists should be podcasting more. So I'm glad that more are doing so now, including you. I mean, as you said, I think, you're an expert, so you can talk really deeply about coronaviruses. So it makes perfect sense for you to do that. By the way, did you know that over here, people are calling you the German Tony Fauci?
No.
That's a pretty good compliment, right?
Yeah, it sounds like a big honor. Yeah.
Yeah. But I don't think I've heard you advise your chancellor, right? But you don't have the same kind of duties as Tony does in Germany, right?
I'm not that close to politics. And in fact, even though I sometimes speak to them, that I'm in the ministries every day. And I don't know. So
let's start by just telling our listeners that you had some experience not only with MERS coronavirus, but before that with SARS-1, right? Tell us what you did with that virus.
Well, at that time, I was with the Institute of Tropical Medicine in Hamburg, and that's, especially at that time, still was something like the reference institution for imported diseases. Mm-hmm. So there was an imported disease, which was a traveler coming back from New York to Frankfurt. He wanted to go back to Singapore, but so for refueling, they at that time they did a stopover in Frankfurt and they took him off the flight because authorities had informed, Frankfurt authorities that there is a SARS patient on board at home in Singapore, and he took a short excursion to New York City. So he was taken off the flight and hospitalized at Frankfurt University Medical Center, and at that time, I got involved to make a diagnosis, but the virus wasn't discovered at that time. So this was like when everybody was fishing around, and there was a lot of talk about one of the paramyxoviruses to be involved, even chlamydia. So what actually the colleagues in Frankfurt did was just inoculate vero cells, and they got a CPE, and I had to go to Frankfurt anyway. Actually for my defense. Mm-hmm. So this was really early on. Yeah. And then I just took back with me a bottle of vero cells in my car. I can't be arrested for that now. It's too long ago. And Yeah. So I just put primer pairs on this, on the supernatant, and got some PCR fragments that I cut out of an agarose gel and put on a nice Sanger capillary sequencer, and came out with some strange coronavirus related sequences that you could discover, translated blast searches that were already available online at that time. And there was not anything close. So it was hits to bovine coronavirus and things. And this is how the story began. We were then in contact with WHO, and labs were actually exchanging their results at that time. And it then became quite clear that CDC, US CDC, had also found other fragments on other parts of coronavirus genomes. And we exchanged these fragments, and then suddenly things started to make sense, because labs could then make long-range PCRs based on each other's random fragments that they found. And it turned out these patients who were actually only indirectly linked epidemiologically, they had the same virus.
And this was at a time when we still hadn't heard officially from China, right, about the outbreak?
Yeah, of course. Nobody knew it. Well, actually, what we know now is that Chinese scientists at that point had already identified the virus, but this hadn't been communicated. But they knew since weeks to months. And
so you were the first to isolate virus outside of China, essentially, right?
Maybe. I think CDC also got it isolated at the same time, and Malik Paris in Hong Kong also got it isolated. It was these three labs.
Yeah. And so since then you have always worked on coronaviruses, right?
Yes, somehow. Yeah.
And you're also involved in isolation of MERS coronavirus, right?
Well, Ron isolated it, Ron Fouché.
Right.
He sent us the virus very early on, and what we did was then to make a diagnostic test and distributed it to public health labs and labs all over the place.
When did you first hear about SARS-CoV-2?
On New Year's Day, I think New Year's Day. So it was like a Victor, a lab member, he informed me about Twitter posts he had seen. This was like information spilled from Chinese Twitter likes. I think already at that early time it mentioned that it is a SARS-related virus, right? And then I somehow heard that Sheng Li is involved, Sheng Li Shi. And this then, of course, made me think, well, maybe there is something to it. Because I know she's working on SARS. I know all her work. And I thought, if she's involved, well, maybe this is somehow popped up during her screening efforts. I know she's looking at population. And later on, it turned out it was really like if I'm correct, if I understood that correctly, it was really something like a good guess on her part for having a coronavirus involved.
When did it first reach Germany?
Well, it came here with the Munich cohort. So this was end of January. I think it was the 20th or 19th of January when a business traveler arrived from Shanghai, actually. And she worked with a car supplier company. So there is a lot of exchange with Chinese companies there. And it was a lady coming there to do things like business consulting, I don't know what. And so it seems that she was mildly symptomatic, but took paracetamol, acetaminophen, and somehow combining her impression of having this subfebrile feeling with jet lag, she just didn't take it seriously and just worked through these meetings and infected 3 to 4 people. So I think it was 4 people she infected in the beginning. And then from there started an outbreak that involved a whole number of people, I think 14 people, if I'm not mistaken. And this outbreak was contained. I'm quite sure it was contained. Even though some people speculate that the virus actually ended up in Italy, but I don't really buy that because this whole thing is completely undersampled. And there are now Chinese viruses that are very similar to the early Munich viruses and the early Italian viruses. And at the same time, there is an additional mutation that came up in the fourth patient in Munich. From that point on was fixed in the whole Munich outbreak. And this didn't turn out to circulate in Italy. So I think it's very unlikely that actually one of the first three patients were the source of the Italian virus.
So according to sequences, this is clearly a bat virus, right?
Yeah.
And what do we know? I mean, I'm surprised there's no more information out of China. Do you think it went directly from a bat, or is there an intermediate as there are in others?
I don't believe in a direct acquisition from bats. I also don't really assume that the Wuhan market was like the point of zoonotic transition. Probably this was just a place where a lot of virus was spread. I think there is now just a new study out in Nature Microbiology, if I'm not mistaken, where they show that there is actually community detection along with influenza surveillance detection already in the first week of January. So it must have been widespread already by that time. So this is a sentinel of just a few hundred influenza samples, and they find it already in the first week of January, and then increasing. So it's not a random hit. It must have been widespread. And probably it was distributed on the market. But I mean, if you consider what happens in markets like this is like people who work on the markets and trade animals, of course, they also go where these animals are bred. And that's where you have contact, right?
But we have no idea what kind of animal it would be, right?
Well, I think there is actually no research published from China except the occasional pangolin. But it's interesting to go back to those early papers describing detection of SARS coronavirus in markets back then. And it was found in civet cats and raccoon dogs. So there was good evidence for findings in both animals. And I don't know why actually civet cats then came into the limelight. And this was like, I don't know, maintained as the narrative, while raccoon dogs haven't been followed up that much. And I haven't seen any data on raccoon dogs now, but I would be interested to learn more about this because this is an industry, right?
Okay. But so it's your feeling that it's got to have come through an intermediate host, right?
That's my feeling, yes. But I have no data, no evidence at all, except the knowledge of those old reports.
And other coronas have done similar things, right?
Yeah, definitely. I mean, the 229E corona came through camels, like the MERS virus. Then OC43 would have come through cattle. There is quite some precedent cases.
A couple of weeks ago, we had Ralph Barrick on, and he was worried that because there's so many human-like ACE2 proteins in various non-human animals, he's worried about this getting into animals somewhere and establishing a new reservoir. Is that a reasonable concern?
Well, there are data already now showing that cats can be infected, right?
Yeah.
The ferret can be infected as a lab model, but then there will be other carnivores. So this doesn't seem unlikely, especially as it seems there is already airborne transmission in these animals.
What is the transmissibility of SARS-CoV-2 compared to SARS? I've seen some studies on R0, which place it below SARS, but it seems to me that it's doing a better job transmitting. What are your thoughts on that?
It's not only the R0, it's also the serial length or the generation time, right? There are different terms for this that describe the length of, let's say, the path the virus has to go from one person to the next person. So how long the generations are apart. And here, this is much shorter, of course. So it took for SARS-1, it took like 10 days or 14 days until the average person in the cohort really was infectious, shedding virus at high concentrations. While for this one, it seems we are speaking about 5 to 6 days. So this is a whole different thing. And this, of course, matches the observation that the virus is really actively replicating in the throat. There's little doubt. So there's really not only the observation that you can easily isolate the virus from the throat from swabs, but also there is subgenomic RNA transcription in throat tissue in swabs without big doubts. And then in one of the cases in the Munich case follow-up study, it's quite interesting. We found there are two different virus populations discriminated by a single nucleotide polymorphism. One population in the lung and the other actively replicating in the throat. And these populations could be discriminated over days in the patient. And then the virus from the throat started the whole rest of the outbreak. So this was transmitted onward, and every patient after this is patient number 4 in the Munich cohort, had then the single nucleotide polymorphism observed in the throat of patient number 4.
And so that makes sense because it's much easier to transmit from the upper tract, right? Because the lower tract, you would have to do really severe coughing and expulsion, I guess, right?
The interesting thing there is, so if you look at the old virus, the original virus present in patients 1, 2, and 3, is the one that ended up in the lung of patient 4. And in the throat, there is the new mutation, and this new mutant is then transmitted onward.
So these mutations are just markers that you use. They don't have any phenotypic meaning.
No, just markers. Yeah,
because there's a lot of misinterpretation of these that they make some basis for the difference, but they're just markers that you can use to track where the virus is going, right?
There is little evidence for phenotypic differentiation now in circulating viruses. I think there is this one observation in Singapore with the deletion of the open reading frame number 8, and then there is a report about some isolates in China that had the polybasic cleavage site deleted in the spike protein.
So let's talk a little bit about transmission. So the virus is in the upper tract. So what are we have heard a lot of things about droplets and contact. What's your impression of what is more important, or are they equally important? Touching someone with contaminated hands or expelling droplets that are rather large?
Well, it's very difficult to quantify. What I found interesting is that there is a recent paper by Christoph Fraser in Science, and he has a compartmentalized model, mathematical model, where he has different, let's say, transmission compartments, part of the whole transmission process. And he estimates contact transmission to contribute about 10% of the overall transmission activity.
And the rest is by droplets.
Yeah, droplets, and as it seems, aerosol as well. This is also a new insight, but if you look at the excretion of the virus, there should be a component of aerosol transmission. There is an interesting study from Singapore. So this is based on air sampling, right? And they sample air, and these air samplers, they make a division, they separate actually the little air droplets that they suck in fractions larger and smaller than 4 to 5 micrometers. So in this study, and this is based on SARS-2, just on a few patients, but they could collect below 4 micrometers, and this is then the aerosol particle size.
Yeah.
And then the other study from Hong Kong in Nature Medicine also, this is also a methodologically well executed study, but not on SARS-2, on other coronaviruses, common cold coronaviruses. They show quite convincingly that there is also a considerable aerosol component. Of course, this is sampling over half an hour. It's integrating a lot of droplets, but probably this is also a realistic exposure time.
Yeah, I mean, I would guess that it's never absolute between droplets and aerosols anyway. Even if it's mainly droplets, there's going to be a small fraction that's in air. And especially if someone is making very high titers, right, in the upper tract, you can imagine that the smaller droplets are going to have more, right?
You know what the CT value in PCR, the first patient we diagnosed in Berlin, a young guy, about, let's say, 22 years old, a student. He was suspected of having an acute influenza infection. He had a CT value of 13.
Wow.
Yeah. So when my lab member from the lab texted this to me, I thought he had made a typo and it's 23, but it was 13. So this is an infectious virus concentration in the throat.
Yeah, it's a lot. So Ian Lipkin has been ill, and he told me his CT was 20.
Yeah, well, that's still in the normal range somehow, right? But something like, I don't know, 13. I mean, I've seen this in camels, in the nose of camels with MERS. There also you have these very high virus concentrations.
Yeah. So that means a lot of virus, so very easy to transmit. Yeah. So what is the situation in Germany today? I noticed you have 113,000 cases. Over here in the U.S., people are using Germany as a model for how to handle this outbreak. Can you summarize what's being done that is working?
Well, there is a lot of diagnostic testing here. I mean, we have a comparably high case number in the charts, but probably if we had done testing at a frequency of that other countries do, our case number would just be smaller, which means that we are actually earlier in the outbreak. And by now, you see case fatality increases, right? So we are now also above 1%, 1.5 something, and it will increase further. So there is this effect that we started to detect very early. So this is like everything started within basically 1 week, where we had the first imported cases, but at the same time also the first detections of bycatch with influenza screening, right? So every lab in Germany is in fact allowed to test if they can, and if they have a quality assurance program in place, they are allowed to test. And that's why so many labs just started testing.
Yeah.
Many labs also started to screen for SARS-2 just along with influenza surveillance. And so some of the first cases were actually discovered along with influenza surveillance. And then, of course, politicians need two criteria. One is TV pictures from other countries, right? Which with an emotional implication, and also, of course, an emotional implication and the expectation that you will have similar pictures at home in your own TV shows. And then at the same time, something like a lab confirmation that the virus is already a domestic problem. It's not something where you can still point the finger at other countries and keep the borders closed, but where you have to acknowledge it is among us. And this happened just immediately in Germany. And so these two criteria were in place. And then I think it took another 1.5 or 2 weeks of discussion before schools were closed. And this is something that didn't happen in other countries, maybe because this second criterion just wasn't in place yet, right? So you saw what was going on, but still you couldn't prove that it's already inside your own country.
So is testing just restricted to people who are ill, or do you expand it to contacts and just to get a sense of who's infected? Do you do more than that?
It's a mixture. So it's still mainly symptoms driven. And I think this should continue. So we should actually continue to test directed by symptoms, because everything else is a waste of resources. But there is a small component of contact based testing, but I think this is decreasing because the public health professionals, they are just overwhelmed by now, and contact tracing with lab follow-up is just very difficult to implement and maintain.
Are you still, the number of cases still rising, or?
So we have a lockdown situation, a social distancing situation, but there are exceptions. And for example, you can easily go out for sports or for taking a walk as long as you're within your family or household context. And it's not like the police is I don't know, billing you and fining you. So it's a soft situation. And well, the first derivative of the incidence is going down, but it's not flat yet.
Is there going to be a role for serological testing in the near future?
Yes, of course. Of course, there will be a role. Especially, I mean, what's now being discussed, of course, is serological testing to qualify workforce to re-enter a normal workforce environment, like in frontline areas in hospitals. And well, I mean, we don't have evidence for immunity based on an ELISA result, but at the same time, we can say this is an infection overcome. And there is good reason to assume immunity, at least for considerable time. Well, I think this is, as soon as capacities are in place, as reagents are being available all over the place, I think it's inevitable that this will start.
So what is the length of the typical disease course, and how long do we shed virus in a way that will transmit it?
Yeah, well, so in an uncomplicated case, this is something we followed up in the Munich cohort. Symptoms are actually restricted to the first week. They improve in the end of week 1. And actually, this is also where infectivity ends if you take cell culture as a surrogate criterion. So what we did is we used 2 different types of cell cultures. One, just plain Vero E6 cells. And the other is Vero E6, but transgenic for TMPRSS2. So for the transmembrane protease that we by now know is used by SARS-2 and to a higher extent than SARS-1. These are cells that Stefan Pöllmann gave us. He made them for his experiments. He's an entry specialist, and for some time, we used these cells in parallel, but actually it turned out that while the TMPRSS2 transgenic cells, they give a better CPE, they are not more sensitive for diagnostic virus isolation. So we just continued using Vero E6 because it's just more handy. And then we did a lot of virus isolation based on swabs and sputum samples. Clinicians knew that we had the intention to do this, so they really gave us very fresh samples. And we did this by 2 labs. So this was done in 2 labs in parallel. And pooling all of these results, we can say that while swabs are PCR positive through the 5th day of disease in PCR, universally, there is no false negative. And sputum samples are actually positive through week 2 or 3 in these uncomplicated cases. So even though they don't really have lung affection, the lung is full of detectable virus. But at the same time, you never isolate virus beyond the first week. So there is never a positive cell culture. And there is also never a positive cell culture below about, let's say, 100,000 RNA copies per milliliter of sputum or per swab sample. So we extrapolate to a whole swab. So this is a criterion, of course, that we are now discussing with clinicians who have to liberate bats as a discharge criterion. And also, let's say, so we have first physicians here who are actually infected, but they feel fine and they ask whether they can go back to work. I tend to tell our boards that they could allow this after the first week because it's not expectable. And I can't tell how it looks for complicated cases. We are only doing this now. And we know, of course, the courses are prolonged and the high virus concentration in the lung is prolonged in these cases. At the same time, it's a bit strange because there is not really a close correlation between virus concentration in the lung and disease severity. There's also not a clear correlation between virus concentration and antibody formation, also between antibody formation and disease, let's say, severity. This is all a bit of a loose correlation that you have. You can make some correlations if you look at many patients, but
yeah. All right. So the key here is that in a more severe cases, and your Munich study was mostly mild cases, right?
Yes.
More severe cases, the kinetics could be different, right?
Yeah.
But it seems to me that, I mean, we can't do infectivity assays in a hospital setting, obviously, but that would be the definitive answer, right?
Well, this would be nice, but it takes a few days.
Yeah.
And many labs have canceled cell culture completely.
Yeah. Sure.
Purposes. Yeah.
And so you can find PCR positivity for a long time after the virus is no longer infectious. That's the bottom line, right?
For weeks after infectivity ends in cell culture.
In the old days before PCR, we didn't know that. We just saw virus infectivity go away, and then we said, that's it.
Yeah.
It's really interesting. And that study, you also looked in stool. And tell us what you found there.
Well, actually, in many patients, the stool concentration is almost synchronous to the sputum concentration. But there is reason to believe that there is active replication in the gut, because there are some patients where you see, okay, the virus concentration is down to 0 in sputum, but still there is excretion of high levels in stool. There is also some patients where it's not synchronous, so there seems to be some active replication. But we never isolated virus in cell culture from stool. Of course, there are explanations there. So the virus could be prematurely processed as it is excreted after having been in this environment of digestive juices for hours. Of course, this does something to the glycoproteins. So it's very well conceivable that this is non-infectious virus after active replication in the gut. This is at least my working model.
Yeah. So you think that virus is swallowed from the sputum basically and gets into the gut?
It is certainly swallowed somehow, but then I think it actively replicates, maybe in the stomach, maybe in the duodenum somewhere. I wouldn't be surprised to find replication there.
You don't think the virus will then traffic from the gut to the lung, do you?
No, I don't expect that.
Mm-hmm. Okay. But in SARS-1, we did see some gut replication, right?
Yes.
And shedding. So it's not unexpected that it would be similar here. What fraction of total cases SARS-CoV-2 do you think there is gut replication?
So what fraction of virus concentration?
No, how many of all the cases of people infected in the lung, how many also have a gut? Do you think it's 100%?
Almost every patient.
Okay, I got it. Do you think it has epidemiological significance, fecal-oral transmission at all?
Well, based on the observation that we could never isolate the virus from stool, and there is also not much evidence from China where, of course, large numbers of patients have been studied. I think I don't expect this to really contribute a lot. Mm-hmm. This is interesting because this was a prevailing idea in SARS-1, actually. There was this Amoy Gardens event that's still completely resolved, whether this was sewage associated or actually aerosol associated. We don't really know.
So let's talk a little bit about ORF8. Mm-hmm. I don't know if you remember, but back in January, I was reading.
You wrote me an email. Yeah, I remember.
I emailed you, because I had been reading your paper where you published not too long ago, I think it was last year, a study on SARS-1 ORF8 deletions and their effect. Mm-hmm. And so I asked you if it was present in SARS-CoV-2. So tell us the relevance of ORF8 in SARS-1.
Yeah, well, ORF8 is this is the gene that had this famous 29 nucleotide deletion. So, which in the zoonotic source, including in civets, but then also in the first human cases, in the first human-to-human transmission chain, this was an intact gene, intact protein. And then the virus acquired a 29 nucleotide deletion that was maintained in the virus lineage that made it into Hong Kong and then started the international outbreak. And then later on in Hong Kong, deletion mutants were observed that had up to 450 or so. I don't exactly remember the number, but large deletions in that area of the genome, in that region. So the whole protein was lost. And we reconstructed this in the SARS-1 genome, so in a reverse genetic context. And we thought actually to observe what was discussed from the beginning, that actually ORF8 is a protein that was only useful while the virus was in the animal reservoir, and therefore could be eliminated after the virus was only present in humans, and so like a civet or bat specific gene. But it turned out this is not the case. So when we reconstructed this, and this was even in the Frankfurt 1 strain, so in a quite late strain of SARS-1, it regained the virus like more than tenfold replication level. So that's actually quite a phenotype. And this was true in cell cultures from all sorts of origins. So we used human cells, but then also bat cells, and cells from other mammals that are completely irrelevant in the natural history of SARS. And in every cell culture we used, there was this gain of replication level through reconstruction of gene 8. And also in, let's say, in human airway, air-liquid interface cell cultures and, epithelial models for the human respiratory tract, the same observation. So you reconstruct this gene and the virus replicates much better. So while we don't really know about the function, we know, of course, it's sitting in the ER. It may be part of the virus particle. This is what we know, but there is little clear knowledge about the function. But it seems to enhance replication. So it's certainly a fitness advantage for the virus. But this advantage has been lost, which is actually something we know in ecology. It's the founder effect, right? So as animals move to an island, if there is no competition for resources, it's fine if they are not as fit as the founder population, as the original population. So the founder population can lose fitness. It only hasn't been described a lot for viruses.
Now, when I wrote you in January, you said that ORF8 is still there, but now recently Linfa Wang in Singapore reported some isolates without it, right? So what does that mean?
Yeah, this is really an interesting observation. So there is a preprint out on that. So the SARS-2 ORF8 also acquired a deletion. And the authors there speculate on an attenuation, which I tend to agree on. I also believe that this should be an attenuating mutant. What I don't really understand from the published preprint is what part of the current Singapore outbreak was caused by this virus, right? Whether this was just a momentary thing or whether actually a large part of the let's say, the paradigmatic Singapore outbreak that was well brought under control was actually caused by this virus, by an attenuated virus. We could speculate. This, of course, would somehow put the interpretation of the intervention, the epidemic intervention in Singapore into a different light.
Yeah, for sure. Do you think this deleted virus will sweep globally?
I think so I read somewhere that similar mutants were also encountered in other parts of Asia, but they never really stayed. So they were only transiently observed. But well, actually, if you look at the sequences available, this is really very undersampled. Even though like GISAID, they are proud of how many sequences they have. If you consider this is really a pandemic now, and how many cases there are, of course, the sampling in terms of sequences is still very, very low. So it's well conceivable that these things keep popping up and keep being spread, and then maybe they are outcompeted by wild type. So I think they should be under some kind of purification. Of course, they are probably eliminated as soon as a competing lineage that is wild type comes into the same population.
And you mentioned before a deletion of this furin site. So let's talk a little about that. So the furin site is unique to this virus compared with the bat precursor, right? Do we know it's used at all for cleavage?
No, we don't really know, but there is some initial evidence, and I mean this is really a main difference between the two viruses. Mm-hmm. Actually, it's also not that it appeared out of nowhere. There is some, if you look at the broader diversity of bat-associated SARS-related coronaviruses, there are some that have an imperfect, let's say, furin cleavage site, polymer cleavage site. So you see that there is some selection into this direction.
So you said that viruses were isolated in Hong Kong lacking the furin site. Is that right?
Was it in Hong Kong? I don't know. I think there was a report from China where such viruses have appeared, but also there they didn't really maintain themselves. And so what's also known is that as you passage the virus, this cleavage site is often lost.
But you had said before that TMPRSS2 is really the protease that's cleaving the spike, right? So maybe there's less of a role for furin?
Yeah, so the TMPRSS2 is priming the site.
Mm-hmm. Okay.
It's certainly not happening at that site, right? It's at a further downstream site.
Yeah.
But yeah, I think the furin site can assist.
Yeah, and so I'm sure those experiments are ongoing to take out the furin site and see what happens, right?
Yeah, of course. Mm-hmm.
Yeah, that would be very interesting.
Yeah.
Is there anything about this outbreak that has surprised you, given what you know about coronaviruses?
Well, I mean, I'm completely surprised that SARS is back with this level of transmissibility, right? Mm. And, I mean, this is an influenza pandemic without any drug or vaccine.
Yeah.
So, this is a little bit more than a surprise, right?
Yeah, for sure. Me too. I agree with that. Yeah. Do you think we should have had drugs that would have worked? I mean, it seems to me we could have, right? If
CEPI had been in place back then, and some more clinical studies, even only phase 1 clinical trials had been done for drugs or vaccines, we would be better off today, I'm sure.
Yeah, I think CEPI is a wonderful model for how drug and vaccine development should go now, right? Because it doesn't depend on profit.
Yeah, well, it is of course a co-financing for many companies. It's only a co-financing, but it's in some cases it's also just a blessing that companies want to have and want to take. It is. It's a pity that it wasn't in place back then.
Yeah, I agree. So what's the end of this? Do you see what has to happen before this pandemic stops?
Yeah, I was, for a few weeks, I was really like, I became a vaccine romantic, and I thought, well, maybe we can really cut everything very short and have a vaccine in autumn or something. Mm-hmm. But the more I speak to people who really know something about vaccines, which, and this doesn't include me, the more I have to acknowledge that it's just not doable. So we will have to wait at least a year and probably somewhat longer. So repurposing, there are some ideas. Of course, it's not only chloroquine. There are some other ideas. Mm-hmm. And there will be more ideas coming. But I mean, this is not very promising.
Yeah.
Lucky. And the other thing then is, I don't know, but I think there is a reason why China used a mobile phone app early on. And there is also some explanation behind this observation that, I mean, if the numbers are correct, which nobody knows, the period of non-lockdown in many parts of China without a resurgence, without reoccurrence of exponential transmission, may be linked to the use of mobile app contact tracing. So the more I thought about this in the last, let's say, 2 weeks, the more it becomes clear to me that if we want to get out of a lockdown situation, we will have to use mobile phones widely. So these tracing apps, I think this brings the granularity in case tracing, contact identification, and the speed that we need. So if you consider that almost half of all the transmission activity happens before onset of symptoms, then of course, and the serial length is that short, it becomes clear that actually when I start to be sick, then the one I have infected is already becoming infectious himself, right? So the next generation then is already starting. So it's not only, so if I'm generation 0, then generation 2 will already start. So this means we have to cut out, so we can't cut out those. The pre-symptomatic transmission events, we can't work on these because we need symptoms as an indicator. So this means for an R that is about 1, we have to eliminate almost all of the symptomatic transmission activity. This means we have to be very, very fast. And this can only happen with some automated processes.
That's interesting. So what fraction of cases never show symptoms? Do we know?
Probably a small fraction.
Yeah.
I think pre-symptomatic and asymptomatic is widely confused.
Yeah.
Asymptomatic, well, I mean, in the Munich cohort, for instance, there was in the end no asymptomatic transmission. Because if you really went back and spoke to the people, they would remember some 2 or 3 days of some sore throat, but they didn't take it seriously.
Yeah. Yeah.
And now all of this talk about, let's say, silent seroconversions, I don't know. We are not seeing this. And especially now, I mean, this is not the time to detect silent seroprevalence, right? The epidemic hasn't even begun. At the same time, we're just moving out of the common cold season. So there is all those cases that have residual IgM from their recent human coronavirus infections, and this will cause cross-reactivity with ELISA tests, especially the IgM positive cases. So the predictive value will, of course, increase as we move into May, June, July. But now, I don't know, all of these ELISA studies that are now starting, if they show something like 3-4% over the known PCR-based background in the population, this is probably contributed by false reactivity. And only later on can ELISA tests really identify something like a population background. But then also these cases, this doesn't mean they are asymptomatic. It just means they haven't been diagnosed when they were infected.
Yeah, as you say, they probably had mild symptoms and they didn't even think of it, right?
Yeah, exactly.
People have a sore throat. They wake up a couple of days, it goes away. Yeah. So currently we have 1.5 million confirmed global cases. What do you think is the actual number of infections? How much more than that?
Globally?
Yeah.
Oh, this is for me almost impossible to estimate because consider what is happening in parts of the world where there is no diagnostics at all, except maybe in the capital, in one lab. And this is really the most worrisome prospect, actually.
Yeah.
If you consider that we haven't seen any outbreak globally that is really free running, that is running free. This hasn't happened. There has been containment measures everywhere, but now the virus is entering into Africa. So this is now, not this month, but next month we will start to begin to see, how these outbreaks look.
Do we understand how much population immunity you would need to curtail an outbreak?
Well, I mean, there are those projections between 60 and even up to 80% of overcome infections.
Yeah.
We still don't know the contribution of children, but probably it's all equally distributed.
Yeah. So I think we're probably a ways away from those numbers, right?
Exactly. And then, I mean, consider what supply of vaccines you can have. I mean, you can have a part of the population of the richest countries covered with vaccine.
Oh, I was interviewed by Moscow TV yesterday, and they asked me that, will the rich people only get the vaccines first? Because, I mean, how can you make billions of doses, right? It's a logistical problem. Even if this mRNA vaccine works, we figured you'd need kilograms of RNA to
Yeah, of course. No, I mean, these are fast vaccines, but not mass vaccines.
Yeah.
So the mass vaccines will be protein vaccines, of course, protein-based vaccines, modified spike proteins with an adjuvant.
But as you say, probably there'll be new things arising, because I'm amazed, and maybe you are, that almost how many labs have turned to working on this problem now. It's kind of enheartening, right?
Yeah, well, but it's still very slow. It's much slower than the virus. And there are really parts of the world where lockdown won't work. We can only hope that, let's say, the more rural parts of Africa, and it starts to become rural as you leave the capital in many countries, that the spread will be much slower there.
Yeah. I just saw an article this morning. He's saying here in the U.S., a lot of rural communities thought they would be spared, but now they're becoming infected.
We see the same in Germany.
Yeah. All right. Well, I have one more question. Will there be SARS 3, 4, 5, 6 in the future?
I wouldn't expect this, because after SARS-2 becomes endemic, we will have good immune protection against SARS-3 if it's within the species, right? But a coronavirus number 3,
okay.
Look at MERS, right? There is a dominating strain on the Arabian Peninsula, and only on the Arabian Peninsula. This has come up in 2014, 15. This is the strain that also started the Korea outbreak. Mm-hmm. And while before 2015, it was like a co-circulation of several phylogenetic lineages of MERS coronavirus, this one has wiped away all the others and has outcompeted all the others. And we have some evidence that it has higher fitness than the previous ones. So also there is phenotypic changes. And this is a virus that is a zoonotic virus, but it's steadily introduced into the human population. So really, MERS, in my view, is something that shouldn't be forgotten.
So it has potential to eventually become a human virus, you think?
Yes.
Is there any cross-reactivity between SARS-1, 2, and MERS?
There is some very limited cross-neutralization between SARS-1 and 2, but nothing at all between SARS and any of the other ones.
Yeah, but of course that's using tissue culture neutralization, which may not reflect what you need for protection, right?
Yeah, of course. I mean, there are certainly cross-reactive T-cell epitopes between all the coronaviruses. That's almost for sure.
All right. That is TWiV special episode. You can find it at microbe.tv/TWiV, any podcast player. You can send your questions and comments to twiv@microbe.tv. If you like what we do, consider supporting us, microbe.tv/contribute. My guest today from Charité Hospital in Berlin, Christian Drosten. Thanks so much, Christian.
Thanks, Vincent. See you.
I'm scheduled to come to a meeting in Germany in October. Do you think that will be canceled?
I don't know. I would be happy to have you here should you come, but I really can't predict.
Yeah,
maybe you have a good mobile phone app and you are a good an elite tracker. They will let you in.
I hope it's okay. I'd love to come visit. Yeah, so if I do go, I was going to go to Heidelberg and visit Matthias Fischer, and I would love to come to Berlin and visit you. So that would be fun. I'm Vincent Racaniello. You can find me at virology.blog. Thanks to ASV and ASM for their support of TWiV. Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.